In this video, we're going to look at the conduction system of the heart. What is stimulating those myocytes to contract so that we can pump blood? So we have the myocytes, or the contractile cells, and then we have excitable cells called the SA node and the AV node. So the SA node is the sinoatrial node, and that is found in the posterior wall of the right atrium. And that is our primary pacemaker. That group of excitable cells is what is going to initiate the action potential that will spread to the muscle cells. We also have an AV node, which is the atrioventricular node, and those cells are also excitable cells. Without the SA node, the AV node could actually stimulate muscle contraction in the ventricles enough to keep the heart functioning. But the SA node is the pacemaker because it depolarizes faster, about 70 beats per minute at rest. And then we have conducting cells. So the bundle of His goes down the interventricular septum, and then at the apex of the heart, we have the Purkinje fibers that will stimulate the ventricles to contract. So excitable cells and conducting cells. So I want to look at the details of how the action potential occurs in the SA node. But let's just have a quick review of how an action potential occurs in a neuron. In a neuron, we have a resting membrane potential. And that was somewhere around minus 70 millivolts. And then if there was a stimulus, and if it depolarized to a threshold level of about minus 55, then an action potential would occur. And we would have a depolarization phase, a repolarization, hyperpolarization, and then go back to our resting membrane potential. When we have the depolarization phase, that was because sodium ions were moving in and changing the inside charge of the cell to positive. The repolarization phase was because potassium ions moved out. And we had excess potassium ions moving out that caused this hyperpolarization phase, and then it went back to a resting membrane potential. In the SA node, this is a little bit different. So now we don't have a resting membrane potential. There is no point when the SA node is at a constant resting voltage. So at about minus 60 millivolts, there is a change in the charge automatically. And the charge is going to gradually, slowly start to depolarize. And then we have a rapid depolarization phase, and then a repolarization phase. And then it will begin again automatically. So all of the channels that are involved here are voltage gated, which means that they are opening or closing in response to a change in the voltage. So this early initial slow depolarization phase happens because sodium channels open when the charge is about minus 60. Those sodium channels open, sodium is going to move in, changing the charge of the SA node cells to positive. As the positive charge reaches this threshold level, which is actually about minus 40 now and not minus 55, that is going to trigger calcium voltage gated channels. Now the calcium voltage gated channels are going to open, and that causes the action potential. Calcium is going to move in, bringing a lot of positive charge in. So that is significantly different from an action potential in a neuron. Calcium does not play a role in the action potential in a neuron. So we have the slow depolarization phase is because of sodium. This is our pacemaker potential. It's like kind of like a graded potential. And then the action potential, depolarization, is because of calcium, and then repolarization is the same. Potassium is going to move out, moving positive charges out, and that causes the repolarization phase. The potassium channels are also voltage gated. They are going to open when the charge of the cell is about plus 20. And this isn't exactly lined up, but it goes from approximately minus 60 to approximately plus 20. In a neuron, it can go higher. It can become more positively charged. So when the charge of the cell is about plus 20, then the potassium channels will open. And then we go back down. Now we don't have a hyperpolarization phase. As soon as the charge of the cell reaches about minus 60, those sodium channels are going to open again. And this is why the heart is autorhythmic. As soon as the charge reaches minus 60, sodium channels automatically open. The voltage-gated sodium channels automatically open. Once it reaches minus 40, then we have the action potential with calcium. Okay, so this is why one of the drugs that we can use to treat high blood pressure are calcium channel blockers. It can slow that depolarization phase. So that is how an action potential in the SA node is different from an action potential in a neuron. Now let's look at how that action potential is going to spread through the heart. So remember, when the heart is contracting, the atria contract together and push blood down into the ventricles, and then the ventricles contract together and push blood from the right side to the lungs and from the left ventricle to the aorta to the rest of the body. So the ventricles are contracting together. So we want the blood to move up and out of the ventricles. So when the action potential occurs in the SA node, it has to spread to the myocytes, the cardiac myocytes. Just like before, the nervous system has to stimulate muscles to contract. And then the muscles also depolarize. So then when the muscle cells depolarize, then they contract. So one thing that is important to remember from our overview video is that we have gap junctions in between cardiac myocyte cells in those intercalated discs. So when depolarization is occurring in the muscle cells, the ions can travel very rapidly through the gap junctions. That is an electrical synapse. Then the cardiac myocytes can contract almost simultaneously. So let's look at how the action potential is going to spread from the SA node to the muscle cells, and then how it spreads down to the ventricles. The SA node is our pacemaker, and that is in the posterior upper portion of the right atrium. That action potential is going to spread throughout both atria. So the electrical connection between cells, because of gap junctions, helps that spread very quickly. So the depolarization of the SA node leads to depolarization of the muscle cells in the atria. Then that signal, that depolarization signal, is going to go to this little spot here, and that is called the AV node. Now the AV node has a very specific function. What it does is it actually slows down the spread of the action potential. And this is important because when the atria contract, we need to prevent the action potential from spreading downward through the ventricles, because that would make the ventricles contract at the same time as the atria. So the AV node slows down that action potential. This is important because then the atria can contract before the ventricles start to contract. So then from the AV node, the signal is going to transfer through this branched structure that moves down the interventricular septum, called the bundle of His. These conducting cells send the action potential down to the apex of the heart, and then it is sent to these fibers, these conducting fibers at the bottom of the heart, called Purkinje fibers. This way, the action potential moves down to the apex and then causes contraction of the ventricles in an upward motion that allows the blood to be pumped up out of the right atrium to the lungs, or the pulmonary circulation, and up out of the left ventricle into the aorta. So the order of excitable to conducting cells is from the SA node to the atria, to the AV node, then to the bundle of His, to the Purkinje fibers, and then to the ventricle myocardial cells. We can look at the electrical signals moving through the heart using an ECG, or an electrocardiogram. And this is where you put electrodes on the skin. And because our body is a lot of water, we are very good conductors of electricity. So the electrical activity in the heart can be conducted through the skin to the electrodes, and then we can look at what is happening on an ECG. So up here, when we have depolarization of the SA node, that is going to stimulate depolarization of the atria. When we depolarize the atrial muscle cells, that produces this P wave. So the P wave is indicating the depolarization, the movement of the ions, and this will lead to the contraction of the atrial muscle cells. Then the signal is going to move from the AV node down through the bundle of His to the Purkinje fibers. That electrical impulse is going to stimulate the ventricles to depolarize and then contract. When the ventricles depolarize, that forms this QRS complex. So this is indicating the depolarization of the ventricle muscle cells. And then this last little wave here is when there is no contraction, so relaxation or diastole. Okay, so excitation, contraction is when we have systole, and relaxation is diastole. So the repolarization of the ventricles is occurring here with the T wave. So in our ECG, we can see the P wave, depolarization of the atria. QRS is depolarization of the ventricles, and the T wave is repolarization of the ventricles. So what's missing? Okay, do the atria just not repolarize? They do, but it occurs during the QRS, so we can't see a specific repolarization wave for the atria. So it is still happening. So the heart contracts automatically. It has autorhythmicity. But what if we need to increase our heart rate? Okay, what if we have a stress response, or what if we're exercising? Then the autonomic nervous system can increase the rate of contraction and the force of contraction. So the autonomic nervous system is the sympathetic autonomic nervous system will release norepinephrine on the SA node, which can increase the rate of depolarization. It will affect the sodium channels so that more sodium can move in and depolarization can happen faster. Okay? Norepinephrine can also stimulate the contractile cells of the ventricles and can increase the force of contraction. Whereas the parasympathetic nervous system can release acetylcholine on the SA node, and it can decrease the heart rate, right? Remember, our parasympathetic nervous system is our rest and digest system, and it releases acetylcholine. The parasympathetic system doesn't directly affect the force of contraction. So the lack of sympathetic stimulation will decrease the force of contraction back down to normal. We can have problems with the conducting system, and that can cause the heart to contract in an irregular way. And so we call this arrhythmias. Arrhythmias are when the atria and the ventricles don't contract properly in the right way. And these can be benign and not severe, or they can be very severe. So when the heart is not efficient, you can start to have symptoms of like not getting enough oxygen. So you would feel short of breath if your heart wasn't pumping blood efficiently. So all kinds of different things can affect the conducting system of the heart. If someone has a heart attack, if they have high blood pressure, if they had a congenital malformation of the heart or the valves, sometimes streptococcus infection can cause rheumatic fever, which can affect the valves. So there's multiple different things that can affect the conducting system. But there's one that is very common, and most often it's just benign, and I want to just mention that quickly. And that's called a PVC, or a premature ventricular contraction. And this is almost like, have you ever felt like your heart skipped a beat? Okay, or you get that little fluttery feeling and then it goes away? Okay, that is almost like your like a muscle cramp, or like when you have the hiccups. It's just a weird thing that can happen where the ventricles might contract a bit too soon. So premature ventricular contraction. And that can give you that heart fluttery feeling. And this is what an ECG would look like if you had PVCs. So here we have our normal P wave, we have our normal QRS, and we have our T wave, and then P, QRS, and T. And then over here, we have this funny thing where the ventricles contract and there's no P wave. So the ventricles just contracted a little bit too soon, and it can give you a funny feeling, and then it usually just goes back to normal. So now what I want to do is I want to compare our ECG, P wave, QRS, T, with what is happening in the left ventricle. We're going to look at the pressure changes in the left ventricle and the volume of blood changes in the left ventricle. We're going to look at the left ventricle because that is what is sending blood to the systemic circulation. So this diagram, I'm just going to warn you, it looks a little scary, but it's not that bad. So this is the whole process, and there's multiple steps, and I'm going to go through each one of these things individually. I'm just showing you this. I'm going to zoom in a little bit more, and I'm going to kind of cut off the top and the bottom a little bit so that we can see what's happening. So we have three things we're going to follow here. One is the pressure in the left ventricle. LVP is left ventricle pressure. Then we're going to look at the left ventricle volume. So the volume in the ventricle is going to fill up, then it's going to empty, then it's going to fill again, and we're going to look at how that is happening related to the ECG. When we have contraction, it is called systole. When we have relaxation, it is called diastole. So here, this LV-EDV, this means left ventricle end diastolic volume. So end diastolic volume means how much blood is in the ventricle after it has been relaxed. Okay, so after being relaxed, this is when it is the most full. And then down here, we have left ventricle end systolic volume. So the end systolic volume is when it is the most empty. But we will see that it is actually never completely empty. Okay, so step one, we have our P wave. The P wave is going to cause depolarization of the atria, which causes the atria to contract. So what is happening with the volume of blood in the left ventricle before and after the atria contract? So the ventricle is actually almost full before the atria even contract. Then when the atria contract, it is completely full, somewhere around 120 milliliters of blood volume in that full ventricle. The end diastolic volume is about 120 mils, and this is at rest. So we have the P wave, causes contraction of the atria, which fills the ventricles. When the little extra blood goes into the ventricles, we increase the blood pressure a little bit. Then we have our QRS. The QRS is when we depolarize the ventricles, and then we begin to cause this increase in pressure in the ventricles. Now, what are these two blue regions? These regions here are called isovolumetric phases. Iso means the same. So this is when the volume of blood in the ventricles is not changing. And that has to do with the valves. In order for blood to move into or out of the ventricles, certain valves have to be open for the volume to change. So over here, at the beginning of our first isovolumetric phase, the P wave occurred, atria contracted, and blood went into the ventricles. Then what had to happen right here is those AV valves have to close. So the blood moves from the atria into the ventricles, then the AV valves close, and that gives us our first heart sound. Now, this isovolumetric phase, all of the valves are closed. But the QRS is going to cause an increase in pressure because now the ventricles are starting to contract. As the pressure increases, it's going to reach a point here where the pressure is going to be high enough to open the semilunar valves. So now, number 9, the semilunar valves open, and this allows the blood to move from the left ventricle into the aorta. Now you can see the volume of blood in the ventricle is decreasing, and that's because the QRS stimulated contraction, the semilunar valve opened, and now the blood is leaving the ventricle. So the increase in pressure causes the semilunar valve to open. Then our end diastolic volume here from about 120 mils, that is going to decrease. Now at the end of the ventricular contraction, we have our second heart sound. Now those semilunar valves are going to close. And this is our second isovolumetric phase. Now, take a look at the amount of blood that is left in the ventricle. What is the end systolic volume? It is about 50 mils. After contraction of the ventricles are never completely empty. There is always a little bit of blood volume left. And then the cycle is going to continue again. So now the ventricles are as empty as they're going to be. And then, in order for them to start filling again, we need to open the AV valves. Once the AV valves open, the ventricles can begin filling again simply because of gravity. Then we're going to have another P wave, and we're going to add that little bit extra when the atria contract. Then the whole process is going to happen again. So most of the ventricle filling is happening before the T wave. So as soon as those AV valves open, blood can start moving down from the atria into the ventricles. So the atria contracting just gives it that little bit of extra volume. And then the QRS, depolarization of the ventricles, causes the ventricles to start contracting, and that pressure causes the semilunar valves to open. So now the last thing is, how do we figure out what the stroke volume is? So the end diastolic volume, when the ventricles are full after relaxation, is about 120 mils. And then after systole, the end systolic volume after contraction, okay, there's about 50 mils left in the ventricle. Okay, so you simply subtract those numbers to get the volume of blood that has been ejected, and that is the stroke volume, which is about 70 mils. So here is a summary of all of the different steps of events that are occurring with the cardiac cycle compared to the electrocardiogram.